Hello, everyone, and good afternoon. I'm Lori Plout-Snyder, the Dean of the School of Kinesiology, and it is my pleasure to welcome you to the can you believe that, 9th annual Katarina Borer Lectureship in Exercise, Endocrinology, and Metabolism. On behalf of our school, I would like to thank our very own Professor Greg Cartee for sharing his time and talent with us today. Sometimes you think that the best expert in the field to give the most interesting talk right on topic might come from another country or another coast or far away, but sometimes you're fortunate enough to have that expert right here with you. And so we're very grateful to Greg for his time today. And I'd also like to express our gratitude to Katarina, Professor Emerita, Katarina Borer, and her husband, Dr. Paul Wenger. Their son joins them here with us today too, for making this lectureship possible. So let's give them all a round of applause. And now I'd like to introduce Professor Emerita Katarina Borer.
So you may find it a little awkward that Katarina Borer is introducing a speaker in Katarina Borer's lectureship, but that's really because Professor Greg Cartee was introducing speakers for the past 8 years. And of course, this time the committee that selects the speakers chose him unanimously and enthusiastically, and so he couldn't do it. And for next year, the new, actually Greg is stepping down as associate research dean this year. So next year, a new one will be Professor Natalie Colabianchi, and so she will be introducing the speakers next year. So I'm going to maybe abuse a little bit this opportunity to tell you just briefly about why there is this speakership and why it doesn't have my name. So the story is that I was not an exercise physiologist or exercise endocrinologist. Let's say in late 1970s when I was basically offered a job in kinesiology. But I did an experiment as a postdoc in neuroscience lab, which basically was my inspiration to go in the direction of hormones. I did a very simple study, and my colleagues will probably say it's simplistic. I wanted to know why a little critter like hamster, as big as your fist, has motivation to run kilometers each night. I mean, they are unbelievable how much they run. And so I wanted to see whether this running was built in, let's say, genetically in the system of energy regulation or weight regulation, so that if the animals grew fatter, running would keep that in check. Well, that hypothesis didn't work at all, and exactly the opposite. When either animals or humans become fatter, they become less motivated to move. That's not a very happy idea. But what happened was that I discovered that free running in these animals at an age when they normally don't grow stimulated a really very significant skeletal growth. They became bigger hamsters. Everything, skull, skeleton, bones in the limbs grew longer. And that was, speak louder, is that what you're saying? Basically, this was, I then became an endocrinologist and developed an assay, a radioimaging assay for hamster growth hormone and prolactin, and sequentially blend them and figured out that the running was stimulating or increasing pulsatile growth hormone secretion. And so that became fascinating part of first few years of my career here in kinesiology. And of course, I learned more exercise physiology, and so I applied my understanding of hormones in how they influence metabolism. So, so over 40 years of doing that, I learned a lot about hormones and exercise endocrinology, but I also was struck by how few exercise physiologists study hormones. They can basically get a lot of mileage and interesting information of just studying physiology and metabolism. And so what basically then happened was that I wrote down what I learned about endocrinology in the two monographs. And then 2 years before retiring, my husband and I decided that we would like to, or especially I would like to, see this discussion and focus on hormones in exercise metabolism perpetuated. And so we have then offered and found for 8 successive years some very accomplished scientists who presented different aspects of hormones and exercise. So I was really asked to do this, to introduce Greg's many accomplishments and put them on a single PowerPoint slide, which is quite a challenge. And basically, Greg has trained as an exercise physiologist and been for 35 years active in the field. And he spent about first 15 years at University of Wisconsin in medicine, and then came to Michigan in 2004. And here he is not only professor of kinesiology, but he's also professor in molecular and integrative physiology, and he is research professor in gerontology, and he's also on mentor for graduate students in additional groups, which is a graduate program in cell and molecular biology and program in biomedical science. So he's really been recognized on this campus as an eminent scholar. And he also acted as an interim dean, and for 11 years he has been assistant research dean and introduced speakers for this talk. What is very impressive is that he trained so many doctoral students. So at the University of Wisconsin, by my count, 14 doctoral students and 18 at Michigan, and many master's students, as you see. I can't use a clicker either. Published 142 papers, and what I want to end with is to say that he also was inspired, just like I was inspired by my hamster study, where exercise stimulated increased pulsatile growth hormone secretion. He was inspired by 2 studies, classical studies, which were of same vintage as his entry in the field of exercise physiology. And the one on the left was by Kurt Richter, right? And it was also in 1989. And he basically did single leg exercise, which demonstrated that it is the contraction in the muscle that increases the action of hormone insulin. Insulin actually rules metabolism after food intake and converts the nutrients into storage molecules, which are inert, glycogen and fat. But when human or animal exercises, the exercising limb, but not the sedentary limb, increases uptake of glucose for metabolic expenditure. That's one of the inspirations. The other one was by Ralph Efronzo, which demonstrated that, again, I'm sorry, I clicked. Okay, which demonstrated that when people become obese and animals become obese, the major deficit in this action stimulated by exercise, which is greater uptake of glucose, increased insulin sensitivity, declines, and they become insulin resistant, which is associated with many other pathologies. And so now I'm going to pass the baton to Greg, who will talk to you about 35 years of this research, pursuing the mechanisms for insulin sensitivity after exercise.
Thank you, and Katarina, thank you for the nice introduction. It's a real honor to be asked to give this talk because I have a lot of respect for Katarina's both accomplishments and her passion for research. There's no one that I know of who has as much passion for research as she does, and she continues to. She's actually quite productive still as an emeritus professor. She continues to publish and to edit publications, and so I'm really honored to have this privilege. So I'm going to go through a series of slides related to some of the work, but I like to start out by acknowledging who did the work instead of waiting at the end when everybody's tired already. So these are the current lab members. The names are the people in the lab. The picture isn't completely up to date, so everybody in the lab isn't represented there, but also I've been here at Michigan. I'm coming up on 21 years here, and so here are some of the people through the time at Michigan who've been here and other collaborators who I've worked with. And I also want to acknowledge the research support from NIH. So my brief outline, what I'm going to talk about is first is to talk about the importance of skeletal muscle for health. And then I'm going to talk about a little bit of background on the effects of acute exercise on insulin sensitivity. So I'm just going to be talking about one session of exercise, not chronic exercise over days or weeks or months or years. And then I'm going to spend a good bit of time talking about a protein known as AS160, some research. We'll get into a little bit of detail on some research that we've done. And then at the end, a short amount of discussion of some very recent research related to the role of glycogen in actually reversing the increase in insulin sensitivity. And then brief conclusions. So glucose homeostasis is very important. Everybody can pick your favorite tissue or organ. They pretty much all are involved in one way or another. We are very interested in skeletal muscle. And I'm going to show you a slide very similar to what Katarina did, which shows this classic study by Ralph DeFranzo, where he did a method that he created called the euglycemic hyperinsulinemic clamp that allows you to measure glucose disposal under conditions of high insulin, like you might have if you had a really big meal. And you can see in the healthy controls that the largest portion of glucose uptake is going into skeletal muscle. So they have catheters in place in this particular experiment. And then when as Katarina showed in individuals with type 2 diabetes, which used to be called adult onset diabetes, there's a large deficit in that insulin-stimulated glucose uptake in the muscle. And so what you can see here is that, in fact, this insulin resistance precedes the development of diabetes. So it's a primary event that has to occur along with some other defects. And currently about 35 million people in the US have type 2 diabetes. It's important to say that a lot of people don't always progress all the way to type 2 diabetes, but they have insulin resistance. And this is essentially what is called prediabetes. It's an insulin-resistant condition. There's currently almost 100 million people in the US with prediabetes. And this confers poor health even in those who never develop diabetes. So for example, there's an increased risk of cardiovascular disease, of some types of cancer, and of cognitive dysfunction. And so there's a lot of problems, even the people that don't go all the way to diabetes. So what is the effect of exercise on insulin sensitivity? Obviously, there's an effect, or I wouldn't be standing here talking. But this is actually the seminal study actually by Erik Richter. Before he did the work in the humans, Erik Richter from Denmark did this classic study in rats. And so the x-axis shows the insulin concentration, and the y-axis is glucose uptake. It's actually muscle glucose uptake. So this was rats. They were either sedentary or they did about 45 minutes of treadmill exercise. And these measurements are made not during the exercise, but about an hour and a half after the exercise. And you can see in the open circles, the increase in glucose uptake as insulin goes up. That's exactly what you would expect. And in the triangles, you can see at any given insulin dose, there's a greater insulin-stimulated glucose uptake. And so there's a leftward shift of the dose response curve. And so that's a classic indication of improved insulin sensitivity. And this is the study that I believe Katarina also shared. This is this very elegant model that the people in Denmark use. It's a one-legged exercise model. And so it's a kicking exercise with one leg, a knee extension, where you isolate pretty much the quadriceps muscle doing the exercise. And you have the ideal control muscle, the sedentary contralateral muscle. So you can compare the person's who has this, obviously the two legs are from the same person with the same genetics. They have the same nutrition. They're exposed to the same hormones, et cetera. So it's a beautiful control experiment. And they did in this experiment, again, a euglycemic hyperinsulinemic clamp. So you see an insulin dose response curve there lifted. In this case, the filled circles represent the sedentary leg. So you see the increase with insulin, but you can also see this leftward shift in the dose response curve, which shows again in humans, there's an improvement in insulin sensitivity. This seems to be a fundamental effect that's been seen in other mammalian models as well. So I'm going to go through a lot of work real quickly that we did through the years, really characterizing the effects of acute exercise on insulin sensitivity in a variety of different conditions or different participants, or et cetera. And so we've looked at different, the research we're going to be talking about is in rodents, my research, but I'm going to talk also about what people have seen when they've studied humans. So in rats of a variety of ages, we've seen an improved insulin sensitivity after one bout of exercise. And people who have done this in humans also see this effect. So it's in fact true in humans. Many of our earlier, in fact, most of our earlier studies were in males, but we've been recently studying females quite a bit. And so we see the effects in both sexes, the improved insulin sensitivity, and this is also seen in humans. Again, we initially did a lot of work with animals with normal insulin sensitivity, but it's even more important to study animals or humans with insulin resistance, because that's the population that would benefit the most from improved insulin sensitivity, and we see benefits in both groups. That's also seen in insulin-resistant humans. With genetics, we've looked at multiple different genetic lines of rats and mice, and in every one that we've studied, we've seen improvements in insulin sensitivity. In humans, when you look at individuals with different ethnicity, it's not only genetics. There's other factors that come into play when you look at anything physiologically, but part of the effect is genetic. Not surprisingly, the largest number of studies have been on white individuals, and many of these studies have shown improved insulin sensitivity. There's multiple studies in Asian populations showing acute exercise can increase insulin sensitivity. I could find only 2 studies in Black individuals looking at acute exercise. I'm not talking about chronic exercise, but acute exercise. One showed an increase in sensitivity and one did not. Interestingly enough, these studies were authored by the same author, who is our own Rebecca Hassan when she was a PhD student. And so in one study, they saw the increase. The one that they didn't, I should point out, in that case, they also studied white individuals and did not see an increase in insulin sensitivity. So it's not necessarily an ethnicity effect, that negative result. It was somewhat of a different experimental design. It's not entirely clear why they had a different result. In the literature, I really couldn't find rigorous studies in Hispanic populations or Native Americans looking at acute exercise and insulin sensitivity. There are studies with chronic exercise, but I could not find this. As you know, skeletal muscle is a heterogeneous tissue. There's different fiber types. I'm going to be talking mostly about the epitrochlearis muscle. You hear more about that muscle. It's predominantly fast twitch, and its fiber type profile coincides very much with the entire fiber type profile of a rat. So the rats are predominantly fast twitch. We see an increase in that muscle, but we've also seen it in the soleus that's predominantly slow twitch. And in the past years, we looked at individual fiber types, and we saw increases in many fiber types. So this effect really seems to be rather comprehensive across different fiber types. Humans, the fiber type differences haven't really been studied per se. It's not so easy, but people have mostly studied the quadriceps muscle. There's one study that looked at insulin sensitivity in the gastric medius, but they didn't really parse out the fiber type effects in humans. And finally, we're going to talk mostly about a swim model. That's the model that we use for exercise. We have done treadmill training. We've also done models of exercise such as ex vivo contraction or in situ contraction. We can see improvements in insulin sensitivity. In humans, people have looked at walking, at running, stair climbing, leg extension, weightlifting, lots of different modes of exercise. So the point here is the post-exercise increase in insulin sensitivity is quite robust. So looking at this from more of a physiologic perspective, if you look at glucose uptake in this sort of on my, I guess it's on your right also, the yellow portion there represents a muscle fiber. So here, and so to get glucose into the fiber, it could be regulated at different sites. So obviously, if we looked during exercise, blood flow would be very high during exercise. And if we looked under those conditions, the supply of glucose would be greatly increased. But remember, we're talking about post-exercise, maybe hours after exercise or even a day after exercise. At that point, blood flow is not elevated. So this is not the regulating factor. And plus, we have looked at the effects of in vivo exercise and then coming back hours later and isolating the muscle and setting it outside the body. So supply of glucose would not be different between the groups, and we see an increase in insulin sensitivity. So when you talk about glucose transport, so glucose is a polar molecule. It won't just simply leak through the lipid bilayer. It has to be transported in by transporter proteins, and GLUT4 is the most important transporter in skeletal muscle. And so to really look at transport, we and others have used 3-O-methyl-D-glucose, which is a non-metabolizable glucose analog, which will enter through the transporter but will not be metabolized. And so we see improvements in accumulation of 3-methyl-glucose. And so this is not metabolism, it's the transport process. We'll talk a little bit more about the transporter protein and how the importance of the localization of that protein. So through the years, people have studied insulin signaling. Actually, the insulin signaling, insulin receptor was discovered in the 1950s, and there was a lot of work in the 60s and 70s on insulin signaling, the receptor itself, and more into the 80s and early 90s. Some of these subsequent signaling molecules were studied. One I'll point out here is called AKT. This is an enzyme that phosphorylates other proteins. And we're going to be talking about a protein called AS160, which stands for AKT substrate of 160 kilodaltons. I'll just call it AS160. And it gets phosphorylated by AKT on multiple sites, amino acids, but especially this serine 588 and threonine 642 are important, and we'll come back and talk about that. What you'll notice is the GLUT4 is always going to be in a membrane. It's not going to just be floating around in solution. So it's in this GSV is the GLUT4 storage vesicle. So under basal conditions, it's largely interior of the cell, but with insulin, you stimulate translocation up. So this is the receptor, the red squiggly line here. I'm sorry, the transporter, the glucose transporter. So now I can get access to the glucose and glucose can come in the cell. So through a lot of years of studying different signaling molecules, many people have looked at acute exercise and have found when we talk about these proximal steps, ranging from insulin receptor binding all the way to AKT activation, prior exercise doesn't seem to enhance those signaling steps. So the effects of acute exercise aren't through those proximal signaling steps. So I put in red a couple of our publications, and you can see other people have studied this. So back in 2003, when AS160 was discovered in fat cells, we wanted to study it in muscle, and we've studied that quite a bit over the last 20 plus years. And we saw that you could have enhanced phosphorylation of this protein. In fact, one of the first papers that we published was started at Wisconsin, and it was published in 2005 here, showing greater AS160 phosphorylation after exercise. So I put in red some of our publications. Other people have seen this. So this is a very appealing potential explanation for more GLUT4 translocation. And in fact, two groups, I know you can't read their names, but there's a couple of different groups here where they've looked in humans and in rats, and you can see greater GLUT4 translocation after acute exercise with insulin. So there's a lot of circumstantial evidence that AS160 might be important for this outcome, but really the evidence is not direct. It's really sort of more circumstantial and not proving that it's true. So a number of years back, I think it's now been over a decade, we created and phenotyped what's called an AS160 knockout rat. So this is a genetic model that doesn't have any AS160 in the rat. So AS160 is not present not only in muscle, but in other tissues. And when I say we created, I actually mean Tom Saunders created, because he was the head of the Transgenic Animal Corps for a number of years. He's retired now here at Michigan. And so he was involved in creating these rats. And Nathan Chi, who's still here at the university, helped us with some phenotyping. So if you want to look at diabetes in an organism, you can give what's called an oral glucose tolerance test. So in this, we have wild type, which are the normal rats. and the knockout rats, which have no AS160. And so these actually are siblings. And you can see the open circles that when you ingest the glucose in the wild type rats, glucose goes up, but comes back down quickly. It goes up more in the knockout rats. They're glucose intolerant. On the other hand, they're not diabetic. They don't have fasting hyperglycemia at the 0 time point. They also did Nathan also did the glucose clamp for us. In this case, a high number is a good thing. So the open circles are the normal wild type rats. So they have greater insulin stimulated glucose uptake than the knockouts. The knockouts are insulin resistant. Okay, so now we've got these rats. The typical exercise or experimental design that we're going to use is going to follow this. We're going to have rats. We're going to do an exercise protocol. We've been using the same protocol for decades. It's actually a swim protocol. Rats are natural swimmers, so no problem. They can swim without a problem. We do 4 times 30 minutes with 5 minutes rest in between, and typically wait a few hours, and then we study the muscle. So we dissect out the epitrochlearis, which is a forelimb muscle, and it has, as I said, the fiber type similar to the average fiber type. It also, because it's a very thin muscle, when we incubate it ex vivo, it can get its nutrients by diffusion because you don't have to worry about the vasculature working. And so the rat will have 2 muscles, and so often we will take one muscle and incubate without insulin, and one muscle will incubate with insulin. And that allows us to see the difference of insulin above the baseline without it. So there'll be 2 or 3 incubation steps depending on the situation. Sometimes we use 3-methyl-glucose that I mentioned before, but in some experiments we use 2-deoxyglucose, which is another glucose analog, and then we can analyze these samples. So I'm going to talk quite a bit about a study that was published 3 years ago with Amy Jing, who was the lead of this project. And this is why we made the knockout rats. We wanted to see if AS160 is essential for the increased insulin sensitivity. So the way I'm representing the results here is what I'm calling delta insulin glucose uptake. So we're taking glucose uptake with insulin and subtracting out the basal glucose uptake without insulin. So that shows us the increase that's caused by insulin. And this is really just a positive control. Remember, the wild type are the normal rats. They have AS160. The ones that did not exercise here, the 3 hours post-exercise, there's a big increase in insulin-stimulated glucose uptake. So in the knockout rats, you can see quite right away that they're quite insulin resistant. They have a very low effect of insulin on glucose uptake, although there is an effect, but there is not an exercise effect that improved insulin sensitivity. So AS160 is essential for this increase in insulin sensitivity. Now, we wanted to know if we could restore AS160 into the muscle, would that rescue the effect? There's 2 reasons to do that. Remember, I said AS160 is not just in muscle, it's actually in other tissues as well. And so we knocked out the AS160 in the entire rat. So it was possible that the knockout was acting outside of that muscle. Another thing is just a good positive control to show if you have a knockout, if you put your protein or interest back in, that you can rescue the effect. And so we collaborated with Dongxing Duan from the University of Missouri and his colleague, Dr. Pan. So they are gene therapy experts. They study muscular dystrophy. So they have a great deal of expertise at getting genes into muscles. So they have a method that's called adeno-associated virus approach that we use to increase rescue AS160 expression in the knockout rats. I'm going to spend a little bit of time on the technique because it's relevant to the experiment, the next several experiments. But remember, the normal rats are called WT wild type, and then we have the AS160 knockout rats. So we anesthetize them, surgically expose the epitrochlearis. It's superficial, it's right underneath the skin, so you can just take the skin back. You can inject with AAV for gene transfer into the wild type rats. We had a control AAV, alkaline phosphatase, that really doesn't affect anything that we're studying. And so we did that. And in the knockout rats, we injected AAV with AS160. So we were just really increasing AS160 only in these 2 small muscles. The rest of the rat doesn't have any AS160. Okay, suture it back. The rats wake up very quickly. They're fine, but no problem. We take about 3 weeks so they can express the AS160. It takes a little while for that to occur. Then we come back and do the experiment with exercise and glucose uptake. I'm not going to show you the data, but we did successfully restore the AS160 with the AAV, so that these values and these muscles were very similar to the control animals. So again, positive control, just showing that the AAV didn't do anything. You have the expected result. This is the exercise effect, 3 hours post-exercise, increased insulin sensitivity. Voila, so we add back the AS160 just into the epitrochleus and we rescue the effect. So that's reassuring that the effect really of the knockout is the lack of AS160 in that muscle. Okay, so now one thing, as I said before, AS160 regulates GLUT4 movement in the cell. And so there's an effect that had been seen in mouse knockout models that they actually have less total GLUT4 protein. So you can see how that might confound our results. If you have the AS160 knockout rats have less GLUT4, we might just be looking at that. That might be the problem. So we wanted to see, in our rats, we also see this. You can see in the wild type versus knockout less GLUT4 protein. So we wanted to see if restoring the GLUT4 in the knockout rats could rescue the effect, even if there's no AS160. So we did another experiment very similar to what you saw before, except this time we just used the vehicle instead of using the alkaline phosphatase because it wasn't necessary. So we injected the wild type rats with a sham surgery. Here we're not injecting AAV AS160, but AAV GLUT4. So these rats still have absolutely no AS160, but we confirmed experimentally that the GLUT4 was rescued in these animals. It's the same as the wild type. So again, the positive control of the sham treatment, we have an exercise effect on insulin sensitivity. And interestingly, you can see this is a little bit higher glucose uptake compared to the previous, if you recall how it looked like, but there's certainly not an exercise effect. So adding back the GLUT4 did not rescue the effect on insulin sensitivity. So the lack of AS160 seems to be directly having the effect. It's not a secondary effect of less GLUT4. All right, so I've mentioned before phosphorylation of AS160. There's a lot of places on this protein that can be phosphorylated. We mentioned two really important sites, serine 588 and threonine 642, that seem to get more phosphorylated in response to insulin after exercise. There's another site called serine 704 that's phosphorylated by an enzyme called AMPK. Well, exercise activates AMPK. And after exercise, you can have greater phosphorylation of serine 704. There's some evidence that might play some role in this post-exercise insulin sensitivity. So we wanted to say, is phosphorylation on these three key sites really important or essential for this increased insulin sensitivity? So now, again, we're using the AAV approach. We're using knockout rats, so we can put in the normal AS160 that can be phosphorylated, as well as a mutated AS160, where the serine, threonine, this serine are mutated to alanine, and so they can now no longer be phosphorylated. And so in this case, we're just doing AS160 knockout rats. We've already shown you that if we put in wild type AS160, that we can rescue the effect of sensitivity. The other muscle, the same rat, we put in the mutant. We experimentally proved that these muscles could not be phosphorylated on the three sites that we were interested in. When we come back and look at the results. So again, this is our positive control. So these are knockout rats, but with wild type AS160, there's a nice exercise effect on insulin sensitivity. We had an unexpected result here, actually. We expected we would completely wipe out the exercise effect. But what you can see is we did get an exercise effect, but it's less than the normal effect. So what we see is that AS160 phosphorylation is important or required for a little more than half of the effect, about 55% of the effect. About 45% of the effect was still here in these animals. So phosphorylation is important, but it's not everything. Everything I've been showing you in those experiments by Amy Jing was in male rats, so it was important to see what happens in female rats. And so Haiyan Wang, who's a research investigator who's currently in the lab, did this series of experiments. So we're going to have the 4 experiments I just showed you in male rats, and we're just going to march through them. I'm going to remind you of what happened in the male rats, and then we're going to see what Haiyan saw in the female rats. So we first have what happens in these knockout rats. We saw in the males, there was no increase in insulin sensitivity. High-end saw very similar results. So in knockout rats with no AS160, in the females, no improvement in insulin sensitivity. So AS160 is essential. So if you remember, we were able to rescue this effect. So in the knockout rats, males, when we added wild-type AS160, we rescued the effect. And saw very similar results in the females. So when she put the AS160 back into the muscle, she rescued the effect. So AS160, yes, it does rescue the effect. If you remember, GLUT4 goes down. That is true in both males and female rats that have no AS160. And we saw that adding GLUT4 and normalizing GLUT4 abundance did not rescue the effect in male rats. And saw very similar results in the female rats. So adding GLUT4 back, did not rescue the effect. So it's really the AS160's absence, not the lower GLUT4 abundance. The final of the 4 experiments up till now, they've all been very similar between the sexes. So remember, we had a somewhat surprising result that only about half of the effect required phosphorylation of AS160 in the males. High-end found actually a different result. In the females, it didn't require phosphorylation of these sites. So that was actually quite surprising. We did not expect to see that. That's why you do the experiment in both sexes, because you don't always see the same results. So most of them were similar. This was a little bit different. Okay, so to come back to this GLUT4 localization, the original cartoon I showed you was kind of simplistic. It was just either or. GLUT4 was either inside the GLUT4 storage vesicles or on the cell surface. Actually, GLUT4 can reside in a lot of different membrane populations inside the cell. And so there's the possibility that effects of exercise might be related to changing GLUT4's localization in other sites. So a researcher, this is Knudsen was the first author, but Thomas Jensen is the senior author. He's also from Copenhagen. They do a lot of work related to this. So I showed you before this one-legged exercise model that they do a lot in Danish studies. So they use this one-legged exercise protocol. They exercised and they waited 4 hours, and then they did a biopsy. This biopsy was done without any change in insulin concentration, so it's low insulin. Now, there's a lot of stuff here. Don't worry too much about all the details. But the key things to point out here is the red bar represents the rested muscle. The green bar represents the post-exercise muscle. And you can see in 2 different sites, the T-tubuli and these GLUT4 storage vesicles, the green bar is higher, is significantly higher. So prior exercise altered where the GLUT4 was living after exercise. So you can imagine that might have some effects. Now, when you hit it with insulin, if you have more GLUT4, and say in the GLUT4 storage vesicles, you might get more GLUT4 translocation. And they actually did a clamp and infused insulin and did a second biopsy with high insulin. And now what they see, if we just focus on this right up here, in the sarcolemma up here at the cell surface, there is more GLUT4 in the exercised muscle. So this would be consistent with what we would expect. There's actually some more in another site as well. So this is interesting. So actually, in the grant that we're doing, we just started this year, we're actually collaborating with Dr. Jensen. So we sent him samples from exercised and sedentary wild type and knockout rats with and without insulin. They said they're going to be doing the analysis in October, so it's November. So we're going to wait a week or 2 before we email them to see what happened. But hopefully they can tell us something really interesting from those results. A couple of other things. AS160 turns out to be more complicated. It's not just phosphorylation, but just real quickly, there's other functional domains on this protein. There's some lysines that can influence AS160's binding to the membrane, so it's possible this is playing a role. So that's one possible factor. There's what's called a calcium calmodulin binding domain. Everybody knows in this room probably that calcium goes up when your muscle contracts, and so it's possible that's having a role. And really the enzyme activity, this RAB-GAP enzyme activity is also potentially relevant. So there's things that might be related to these other functional domains that are affecting what we're studying that we haven't studied yet. Another thing to point out is yet another group of Danish researchers in Copenhagen have looked at AS160 binding to other proteins and have shown AS160 binds lots of other proteins, and some of this is insulin regulated. So you could imagine AS160 might be acting because of its binding to other proteins. So there's a lot of different levels that still could be studied in this context. Just quickly to go through the similarities and differences between females and males, I think it's important to say that most of the results that we saw were quite similar between the sexes, but there was one important difference. Please remember that in the normal rats, that you could have improved insulin sensitivity in both sexes. The results were very similar in the 4 experiments in the knockout rats, except that the phosphorylation was important in the male rats, not in the female. So if you think about trying to understand this, what's the underlying reason for sex differences? It could be gonadal hormones. It could be chromosomal differences, XX versus XY. So what we've decided to do in the grant that we've just started is we're going to focus on the estrogen receptor. Estrogen has a lot of metabolic effects. So Hyan is actually leading this project as well, and she's just about at the point to send something down to our collaborators in Missouri. So we're going to make an AAV that we can reduce the amount of the estrogen receptor in the muscle. And then we're going to come back and see if that alters our outcome and our results. Last topic, which is going to be pretty short, so hang in there. We've been talking about the mechanisms that improve insulin sensitivity, but I also have already mentioned is what are if you exercise once, you're not more insulin sensitive for life. Okay, it's a transient event, but it can last a day or two under certain circumstances. So what are the mechanisms that reverse the improvement in insulin sensitivity? And you can imagine that might be important, especially in an insulin-resistant condition. If we could get the effect of insulin sensitivity to last longer, that would presumably be a good thing. Okay, and so again, not under all circumstances, but sometimes the effects can last for a day or two after the exercise. And this reversal of the effect tends to be accelerated when you eat high carbohydrate meals after exercise. And sort of the conventional wisdom about this is that the reversal of insulin sensitivity is related to attaining a very high level of muscle glycogen, which is sometimes called glycogen supercompensation. And this would seem to make sense teleologically. If you exercise and you deplete your energy stores, your glycogen, for the fight or flight circumstances, you'd like to be able to replenish your glycogen in your muscle. You'd want to prefer glucose going to muscle glycogen, so the next time you need to either fight or flight, you'd be able to do it. And so this has been an idea in the literature and just sort of a cartoon presentation of this. So there's a normal amount of glycogen in the muscle. When you exercise, you can reduce that quite a bit. And you can sometimes have improved insulin sensitivity. And it's true that when you eat a lot of carbohydrate, you can get this high muscle glycogen, higher than even the normal glycogen. It's called glycogen supercompensation. And the idea is that somehow this high glycogen might be feeding back and reducing the increase in insulin sensitivity. And actually the work that sort of triggered this hypothesis was work I did 35 years ago when I was a postdoc. Actually, I did it probably 38 years ago. It was published 35 years ago. It took a couple of years getting all the work done. And so this is just that work that was published in 1989. And this is before the knockout rats. It's before anybody knew what AS160 was. And so at this point, these are just normal rats. And if we focus on these gray bars with insulin, you see a certain amount of glucose uptake in the sedentary muscle and a lot more in the exercise. So you've seen that in a lot of my slides. What we saw is this was in animals that were not refed after exercise, so they're fasted. When we refed the animals rat chow, which is about 60% carbohydrate, we eliminated this improvement in insulin sensitivity. It's not surprising if you're not refed, you have low glycogen. When you refeed, you get high muscle glycogen. So we had thought about this for years because that's really, again, just a coincidence, right? They go together, but it doesn't prove cause and effect. But it's actually very hard to manipulate glycogen in the muscle without manipulating a lot of other things. And so once we started doing some of these AAV experiments, we thought, well, maybe we can use this approach to manipulate glycogen. And what we're deciding to do is the enzyme that's rate limiting for glycogen synthesis in muscle is called glycogen synthase 1, GS1. And so we used AAV with what's called short hairpin RNA, which can be used to reduce the abundance of a protein. Okay, so we wanted to lower the amount of glycogen synthase to see what would happen. So some of you saw in the summer, Song-Eun Kwak, who was defending his dissertation, led this project. And I'll remind you, and also for the other people that didn't see that, the way his experiment worked is these are just normal rats. This has nothing to do with AS160 knockouts. In one muscle, we injected the shRNA to reduce glycogen synthase 1. In the other one, we have just a scrambled control, which has normal glycogen synthase 1. So we can look at what are the predicted outcomes in the two muscles. The exercise, so now we have a control where the same rat has got the two different genetic situations. So everything else is the same. We would expect glycogen depletion in both muscles. We did a high carbohydrate, that is chow, just refed them for different periods of time. We had some that weren't refed. We would certainly expect in the normal condition that you would reach high muscle glycogen by 6 hours post-exercise, and you would expect to reverse the insulin sensitivity. We would predict with less glycogen synthase 1, you would have lower glycogen accumulation. So you're not going to get nearly as much glycogen. And the hypothesis really is that we'll keep the insulin sensitivity up higher and longer. That's the idea. So we've broken, we've shown that the glycogen resynthesis may be important. So the key results here, so we got maize and blue for everybody. So we got the blue for the not refed group. This is 6 hours after exercise. To point out in these adjacent bars, the pale bar is the normal control with normal glycogen synthase 1. The darker bar is the muscle with less glycogen synthase 1. And so you can see even in the sedentary condition, there's a little bit lower glycogen in the one with less glycogen synthase 1. But importantly, after exercise, all of them went down, the glycogen went down a great deal. So glycogen is quite low after exercise. When we refeed for 6 hours, we look over here in the refed group in the yellow, and the one with the control amount of glycogen synthase 1, you can see a large increase in glycogen. And in fact, the glycogen has reached a level that we would consider supercompensated. So this is much above the typical level of glycogen. The second bar, you can see there is glycogen resynthesis. There's not zero glycogen synthase 1, but it's much lower. So it's about half as much glycogen resynthesis in this condition. And now let's look at the glucose uptake. There's a lot of stuff here. We're just going to focus first on the blue, the not refed, and just focus on these last two bars. So these last two bars are with insulin post-exercise. And you can see there's an exercise increase, and that's true in both of the muscles. And that's what you would expect. This is after exercise, they're not refed, they have low glycogen. So that's kind of a positive control. The important part is this yellow part with refeeding. And so the pale yellow is the normal control, and you can see there's no longer an exercise effect. It's not significantly higher than its control over here, and there's a diet-induced decrease. But that's also true in the muscle with low glycogen synthase and much lower glycogen. And so we didn't support the hypothesis. Refeeding eliminated the exercise effect on glucose uptake in both muscles. And reversal of this glucose uptake increase did not coincide with the high muscle glycogen. So the effects we're seeing don't seem to be explained that you have to get up to a very high level of glycogen to reverse the effect. So the interpretation, we have to come up with some alternative hypotheses since our hypothesis was not supported. We know if you exercise and then afterward have a high carbohydrate diet, your glucose and insulin in your blood will go up to a high level, and you'll have a high amount of GLUT4 after exercise in the surface membrane. And so a lot of glucose is going to enter the cell. Now we've talked about the priority or the preference to synthesize glycogen, but glucose has a lot of other metabolic fates in the muscle, including glycolysis, the hexosamine pathway, the pentose phosphate pathway, and the methyl glyoxyl pathway. We're going to focus on the hexosamine pathway because there's a lot of evidence that higher flux through this pathway can lead to reduced insulin-stimulated glucose uptake. So our working hypothesis, we're trying to get some funding to study, is that this will have a negative feedback and it will be important for reversing the increase in insulin sensitivity. Other future aims, the work I showed you that Song-Un did was in males. We need to study females to see if the result is similar. And it's a little bit of a semi-translational effect. The experiments I showed were either fed high carbohydrate or not fed. We're going to do more of a graded carbohydrate feeding. So 60% carbohydrate, 30% carbohydrate, 15% carbohydrate to see if we can get a sustained increase in sensitivity with a low carbohydrate feeding. So quickly, next to last slide, overall summary. I hope I've convinced you that insulin resistance is a prevalent health problem and that acute exercise can improve insulin sensitivity in both sexes. That AS160 expression is important for this outcome in both sexes, but phosphorylation on certain sites is important in males only. And this carbohydrate diet-related reversal of the effect doesn't seem to be directly attributable to getting a very high muscle glycogen concentration. So the pursuit continues. We'll be working on this next week, and hopefully we'll have more data in the next year. I want to again recognize the people from the lab through the years. Thank you for your attention. I'd be happy to answer any questions. Katarina, do you have a question? No? Okay, you can talk to me during the reception. We'll talk in the reception. Yeah.
So great work. Presentation. So in the your AS160 knockout, I understand right that the GLUT4 that reduces GLUT4 abundance. What do you think is causing that?
Okay, so it's not because of less synthesis of GLUT4, it's greater degradation. So what seems to happen is GLUT4 actually tends to restrain AS160 restrains GLUT4. If it's not there, the GLUT4 is cycling through the different cycles and to the different locations, including the lysosome where it's degraded. So that's what it seems to be going on. Other questions? Yes, Chase.
In the female animals, where you intend to manipulate the estrogen receptor, is the hypothesis that related to fat oxidation in the female animals and the kind of the known evidence that fat oxidation is a little bit higher in females with exercise? Do you have a hypothesis that kind of around that?
To be honest, don't know what's mediating this effect. There's a lot of possible things. We know that the estrogen receptor, if you knock it out completely, you get a pretty pathological condition. So we don't want to knock it out. That's like with the glycogen synthase. We're going to reduce the amount of the estrogen receptor. So the simple-minded hypothesis is it'll make the females look a little bit more like males, and so that maybe it'll require phosphorylation of AS160. How that's actually, first we have to see, is that what we see? If we don't see that, then in a way it doesn't matter, I guess. But if we do, then we'd have to pursue that. And so whether that would be through changing fuel use or something else, we'd have to pursue that. So that's sort of our thinking about that. I want that answer your question.
Yes, it does. Yeah, thanks.
Any other questions? Yes.
Did you have you seen any direct interaction between AS160 and GLUT4, or is there going to be another signal transduction?
To the best of we have not tested that ourselves. People that have tested it, the AS160 does seem to interact with the GLUT4 storage vesicles, so not necessarily the protein-to-protein interaction. I'm pretty sure when they did the proteomics, I don't think GLUT4 was a binding partner, so I don't think there's evidence of a direct. But the fact that it's localized, so the AS160 is affecting the RAF proteins that affect vesicular trafficking, so it's more as an indirect thing. So the GLUT4 is very hydrophobic. It has to be, it's got 13 membrane spanning regions, so it's got to be in a membrane. And so it's AS160 is going to interact with the membranes that are associated with AS160 that might affect its localization. Other questions? Yeah, Abby.
of your female mouse or studies, or rat, sorry. Do you study them at any specific phase in their estrous cycle? Are they all in the same standardized phase?
Yeah, we have not tried to line that up. You know, we're expecting someone's going to ask us to do that. So there's ways you could do that. You know, potentially there's ways that you can, it's actually called the estrous cycle in rats instead of the menstrual cycle, but I think it's a week long, I forget. So there's the possibility. What I can say, and of course, when rats are co-housed, they tend to get in the same cycle. But we do experiments on different days of the week, and we see the effect regardless. I actually don't think that's driving what we're looking at. I think it's not so much the cyclic as more the chronic changes in hormones, if that's involved. But, you know, it's a good question. It's part of the reason people say, oh, I'm not going to study females, because it might be because that they're different, you know, it's complicated to study or something. But it's an important question, and we haven't tried to.
And then can I follow up one more on that?
Yes.
Have any of your rats been pregnant or had babies or anything like that?
Yeah, these rats are not pregnant. You know, we separate the males and females, so we make sure that doesn't happen. We've done one pregnancy study a long time ago, but it was not in the context of this. It was more about the mother becoming insulin resistant, which is a normal part of pregnancy. And we studied that, but we haven't done it in this context.
So I wonder if it'd be different before and after pregnancy, as there's a big flux in estrogen and.
Yeah, absolutely. Yeah, there's a lot of possible, very interesting things related to that.
So I do have one question. We have the news from our.
Sure.
About the Franzo study, I mean, what happens in obesity? What happens with age, increased age and insulin resistance? You have been pretty much using medium age.
Right.
Stuff like that.
So we've studied older rats as old as a couple of years old, and the exercise improves insulin sensitivity in them. We've studied a high-fat feeding that makes them gain. They don't become super fat, but they are getting fatter, and they're certainly insulin resistant, and we see an improvement in sensitivity in them. Other people have looked at Zucker rats, which are really fat, and they get improvement. So it really seems that you can, what I will say is that when we looked at this, the animals that are insulin resistant, say, here's the normal insulin sensitivity, and here's the insulin resistant. And so if you exercise the normal group, they go up higher. And the insulin resistant group doesn't come up here and match here. They sort of match the sedentary value. So there seems to be a ceiling effect for the improvement, and at least in our hands for the experiments we've.
And in humans, that it's been done in humans?
I don't think that that study has been done exactly in humans. But I think what I can say is when people have looked at very insulin resistant humans, if you exercise once, which is what we're doing, you don't suddenly make their insulin sensitivity up to the level of a healthy person necessarily, but you can get it much closer to that. I've got a question here and bring back to you, Jeff.
With the hypothesized estrogen relation in male rats, would there be a way to test it in male rats by increasing estrogen receptors and estrogen and then use wild type females as a control?
Yeah, so I mean, I think ultimately what we're going to first do the females, because if we don't see a difference in the females, but we really are sort of obligated to look at the males. And to begin with, to look at even reducing the estrogen receptors. Male rats have estrogen receptors. And it's a little confusing in the literature whether they're different amounts or not in males and females. We've seen some say yes and some say no. So we'll try to look at that ourselves. But yeah, we could come back and manipulate and go in the other direction, higher estrogen receptor. And of course, we can come in with testosterone. There's a lot of different options. And there's more than one estrogen receptor. The estrogen receptor alpha seems to be the most important metabolically, at least in skeletal muscle.
I was gonna ask about beta.
Yes, so beta is present in muscle.
The effects are opposite.
Yeah, so it's, yeah. So we're going to start with the estrogen receptor alpha. Jeff, did you have another question?
Katarina's earlier question, that in the insulin resistant animals, so an obese animal and an older animal, is there reason to believe that the mechanisms are that you're looking at in these healthier, younger animals are consistent, or are there other superimposing mechanisms that may be involved with this acute response?
Yeah, so I mean, in a way, yes, there's a lot of layers to that. What's going on here? And to be honest, we have tried to look at some things that are believed to cause the insulin resistance, and we don't see those reversing. So I think the exercise is more compensating for the insulin resistance as opposed to reversing. Again, we're not talking about chronic, we're not talking about changing the body composition or any of those. So I actually think we're doing, and that's what we're seeing as sort of a compensation, not a reversal. And I think they might be through similar mechanisms. I'm not sure what's that ceiling effect. They have the same amount of glucose.
Sorry, I.
Yeah. Okay.
What about the timing? Why are you doing studies 3 hours after the end of exercise? And what happens in between? And why is that crucial in any way?
Yeah, of course, it's always crucial, but there's a lot of different reasons. The 3 is partly convenience. If you look right after exercise, there's a huge effect on glucose uptake without insulin, and that kind of obscures everything. And so that effect needs to wear off. And it takes about an hour and a half to 2 hours for that to wear off. So partly is to get beyond that, 3 hours. And 3 hours, the students didn't want to stay till 6 hours if they don't have to. So 3 hours is, we see a good effect. But we saw the 6 hours. We've actually done 18 hours. We've done 27 hours. We've done 48 hours. So we've done different times. So in our experiments, if you don't feed them carbohydrate, the effect can last. We stopped at 48 hours. So it can last a long time under our circumstances. Okay. Susan.
Greg, is there ever a stress response in these animals that could affect the results just by how they're housed, et cetera?
Yeah, sure. I mean, stress could be an effect. The swimming, what I can tell you is the swimming is a stress. So, but we have looked at other muscles that aren't recruited and we don't see the increased sensitivity. If it was just purely a stress response in terms of the swimming, we would see it in the other muscles. We typically, for some of the experiments, they're singly housed for certain reasons, but we have looked at group housed and we see the effects. So there's certainly always the possibility you're putting on top of it other things, so there could be an effect of stress.
And single fiber research has expanded this or added an angle?
It adds a lot of work, I can tell you that. Students weren't real thrilled about the single fiber work. It's extremely tedious and hard work. So it actually, I started to put some in. I already had gone long enough. I won't put it up. But we did see interesting results. But I think it added to it. And it's just that there's so much to be done. We decided sometimes to go for the low-hanging fruit and not do the hardest possible thing. I think there was a question I saw a hand come up. Is there another hand? Oh, yes.
This activity appears to be sort of a mixture between aerobic and load bearing. I'm just curious if you think the same result would obtain load bearing exercise versus aerobic exercise, what differences you may expect.
Yeah, so we've done the swimming. So again, that's a different kind of exercise, but we have done treadmill running and people have done that. And you see the effects there, and people in humans have done with weight training, et cetera. So it's a pretty promiscuous thing with exercise. A lot of different types of exercise can have the effect. Any other questions? Well, I think, should I dismiss, or I let the dean dismiss, or don't go away, but.
I hope you could join me in thanking Greg for his great presentation. And we will have a reception upstairs on the 4th floor. For those of you who are visitors, there are stairs and elevators right outside this door, and you can ask Greg more questions or just have some fun chatting with each other. So thanks a lot for joining us today.